Good evening, epidemiologists. We are doing our first before-class webinar on observational and experimental studies, and I am recording it for the first time, so let's see how it goes. We are going to review, let me just go to the next slide. We're going to review the following types of observational studies: case control studies, prospective and retrospective cohort studies, and cross-sectional studies. You should also be able to describe the following types of experimental studies after seeing this: randomized controlled trials and quasi-experimental trials. And we'll also talk about associating correct measures of effect with each type of study, for example, odds ratios and rate ratios. This information is also covered in, I think it's chapter 3 or 4 of your book, and it should correspond pretty closely with that chapter. So a quick note on observational versus experimental studies. With observational studies, you're basically collecting data on phenomena that would be occurring naturally anyway. For example, if you tracked dietary information and disease outcomes in a bunch of people with diabetes, but you didn't actually do anything to them or ask them to behave in a different way. In an experimental trial, you're actually imposing artificial conditions on a group of people, so asking them to do things that they would not normally do, and then measuring their response to those things. So if you had the same bunch of diabetic patients and you had said to half of them, okay, you guys eat whatever you want, and the other half, you guys are going to be on this special low-carb diet that we think is going to help your diabetes. And then you track the two groups of people and measure their health outcomes. That would be an experimental trial. A couple notes about observational studies. You would tend to do them when you don't know very much about a potential association, and so it wouldn't really be ethical to do a randomized controlled trial. For example, if you're interested in thinking about the effect of various risk factors on a disease about which not much is known, it wouldn't really be fair to pick one of those risk factors out of thin air and say, okay, these people are going to take vitamin D and these people aren't, because we don't really know enough about that to know that it's safe to assign both of those groups to take it or not take it, and also we don't have any clue whether it would make a difference. So usually you do observational studies first to get a sense about what associations are important. And when you have a little bit more information, you can do a randomized controlled trial. We'll talk more about research ethics later in this course. It's also helpful to do observational studies when you want to study multiple potential exposures and you're not sure which one is going to be the most important one. And also, if there's a natural experiment going on in society, then that's a nice time to do an observational study. For example, right now, vitamin D is very hot and a lot of people are taking it for a variety of conditions. It might be interesting to follow them over time and see what happens to them. In the 80s and 90s, when I was in medical school, there were a bunch of women taking long-term hormone replacement therapy after menopause because people thought that it would keep them younger for longer and prevent MIs, and it was interesting to sort of follow them and see what really happened. We'll talk more about that later. There's 3 types of observational studies that you should know for the purposes of this class. The first one is case control. The second one is cohort studies, and there's prospective and retrospective cohorts that we'll talk about, and also just a cross-sectional analysis of existing data. Case control studies compare 2 groups of people. One group has the disease or outcome of interest, and they are called the cases. The other group does not, and they are called the controls. The controls, this is the important thing about case control studies, the controls must be selected from the population that gave rise to the cases. In other words, they are similar to the cases in every way, except that they do not have the outcome. We'll talk about this more later, but this is like the most important thing about case control studies. And then those 2 groups, that you do a look back and compare them on one or more potential exposures. For example, if you wanted to look at rates of tobacco use in cases versus controls when the cases have renal failure. By the way, there's, I don't think, any data equating tobacco use with renal failure, so I'm just throwing it out there as sort of an association we know nothing about. This is particularly useful in situations where the outcome is rare, and there's 2 reasons for that. One of them is a statistical reason that we'll talk about more later, but when the rate ratio approximates the odds ratio, don't worry about that for now. But the other reason is that, as you can imagine, if you simply followed a cohort of, say, 1,000 or 10,000 people, the number of people that would turn out to have the rare disease that you're interested in would be so small that it would be very hard to compare that number to the number of people who didn't have it. So you'd be better served, for example, if you wanted to look for people who had sickle cell anemia, which is relatively common at BMC because of our racial ethnic breakdown, but say you were looking at people in Nebraska and you followed 10,000 people and it turned out that, 15 of them had sickle cell. You couldn't really compare that 15 with the other 9,000 985 or whatever, and get any kind of meaningful statistical associations because the numbers are so different. But if you went to a hospital, say in Atlanta, that took care of a large number of sickle cell patients and got all of your cases there, and then you got all of your controls from another local hospital or some other place, that would be a great way to get a large number of patients with sickle cell all in one place. And so this is just a logistical issue, but when the outcome is rare, you have to go someplace where you can get a bunch of cases all at once. And that's why these kinds of studies are helpful. So here's an example of a case control study. This is a completely bogus research question. You're interested in whether exposure to firecrackers while pregnant is associated with ventricular septal defects, or VSDs, in babies. This is when there's a hole between the right and left ventricles, so communication between the two ventricles. This is, as heart defects go, this is probably the most common one, but heart defects in general in children are not common, and so it would be very hard in the general population to find more than a couple of people who've had a VSD. But if you go to the pediatric cardiology clinic at BMC, say you could find 100 kids with VSDs. And if you went to the general pediatric primary care clinic at BMC, you could find a whole bunch of children without VSDs. It's actually quite common in case control studies to over-enroll the controls. And you could do it at a ratio of 2 to 1, 3 to 1, or 4 to 1. It doesn't add anything to do more than 4 to 1. And it just improves power, which is the chances of finding a statistically significant result. So a lot of times in case control studies, you'll see more controls than cases, and that's very common. So here are the numbers on your firecracker study. On the top is the outcome, on the left is the exposure. That's sort of the typical way to have it. So you can see that there's 100 kids with VSD on 400 with no VSD. And of those, the numbers are, as you see, those exposed to firecrackers and those not exposed. Here's how you calculate the results in a case control study. Same table, cases, controls, exposure, no exposure, A, B, C, and D. You use an odds ratio, which is calculated as A times D over B times C. So it's just like you just draw an X in the table and you multiply and you're good. And just remember that the A, or the cases exposed, needs to be on the top, on the numerator. So in the firecracker study, the odds ratio was 2.3. Which means, again, this is a made-up study. They're all made-up studies in this class, by the way. This means that if your mother was exposed to firecrackers while she was pregnant, you are 2.3 times more likely to have a VSD than a child whose mother was not exposed to firecrackers when she was pregnant. So odds ratios greater than 1 mean that the exposure is associated with the outcome. It doesn't necessarily mean that it causes it. We're just looking at associations at this point. The key points to remember in case control studies is that they're really only useful when the outcome is rare, and that's because of a thing about odds ratios that we'll talk about later. The selection of cases and controls is really critical. So the main potential for bias in case control studies is selecting the wrong controls, that they're not actually drawn from the population that gave rise to the cases, but they're fundamentally different from the cases in some way. They're healthier, they're younger, they live in a different geographical area. And the problem is that that way might affect their likelihood of having the exposure. So, for example, if exposure to firecrackers is your concern and your cases are 100 kids with VSD from the pediatric cardiology clinic at BMC, and your controls are a bunch of kids who live on, I don't know, an Air Force base or someplace where there's lots of loud noises, right? You're picking two different groups that may have different exposure to firecrackers. So you want to pick people who live in the same geographic area and are the same in many ways. This is the key thing with case control studies. That's really hard. Moving on to cohort studies. So in prospective cohort studies, which is kind of the classical kind, are when a large group of people are selected and followed over time. And then those who eventually get the outcome are compared to those who do not get the outcome. You remember in the first class we talked about person time, and I showed that graph of like 5 or 6 guys that we followed, and some of them had an MI, and some of them moved away or died of something else, and some of them we followed the whole time and they didn't have an MI. Cohort studies are where person time is important. So generally people are tracked over time. And if you're following, 10,000 people, it's not as simple as 100 of them get breast cancer and 9,900 of them don't. Those numbers wouldn't be right for breast cancer anyway, but it's not as simple as X number get it and the total number minus X don't get it, because you're not accounting for people who die of something else or move away or whatever. So that's why person time is important. And it allows you to count time for all the people in the study and not make someone drop out if they get some other outcome. You can still follow them for the time that they were in. So classically in a cohort study, people are compared on one exposure, but exposures can also be evaluated easily, and often are in a cohort study. What happens is that typically in a cohort study, people are given a series of questionnaires over time and asked about a variety of exposures. And you can look at lots of them, particularly if you have a lot of people in the study. The only concern is this thing called multiple comparisons, which is a statistical term that refers to the likelihood that if you look at enough potential associations, at least one of them is going to turn up to be positive and statistically significant. And so it's really a question of how many associations you're looking at by sort of how big your study is. So here's an example of a cohort study. This is a real study, but I made up the numbers. So you want to know whether taking hormone replacement therapy after menopause prevents heart disease, or at least is associated with a lower risk of heart attack. We don't really know whether something prevents or causes. So you decide to follow a cohort of 10,000 postmenopausal women for 10 years. 3,500 of them take hormone replacement therapy. It's their choice. This is a natural experiment. You're not telling them to do it. And 6,500 women of them don't take it. And you follow them for, you can see the total of 33,500 person years, because some of them drop out or whatever. And then of the 6,500 women, they're also followed for something less than 65,000 person years. And they also ask them about aspirin use and cholesterol medication, because that's also of interest. Here's your results from your cohort study. You can see on the left the number of person years, or PYs, that each group was followed for is there. MI versus no MI. You know that MI means myocardial infarction, which means heart attack. I'm here to teach you a few clinical pearls as well as epidemiology. So the rate of MI in the hormone replacement group was 500 per 33,500 person years, which I calculated for you using a calculator, so I'm fairly sure it's right. And the rate of MI in the non-HRT group is that. So the rate ratio is the rate in the first group over the rate in the second group, which is 0.87. As with an odds ratio, if the rate ratio is over 1, that means the exposure is associated with the outcome. So hormone replacement therapy would be associated with MI. If the rate ratio is less than 1, then it's negatively associated, which means that how you would describe these results is you would say women who took hormone replacement therapy were had 0.87 times the rate of MI compared with women who did not take hormone replacement therapy, or they were 13% less likely to have a heart attack than women who did not take hormone replacement therapy. This is described as a rate ratio because in a cohort study, you're really looking at an entire population, including not only people who had the MI and people who didn't, but a whole bunch of other people who, of the people who didn't have the MI, some of them had other things happen to them and dropped out after time. So it allows you, the rate ratio is what you use when you sort of have the whole population in the denominator. It's used for cohort studies basically only. In a retrospective cohort study, it's very much the same idea, but instead the population is followed as a look back. So they're recruited at one point in time, and then the exposures and outcomes have already occurred by the time the study begins, and they are simply asked to recall data over time, or you recall it from the medical record. So for example, you're interested in whether people who take vitamin D are less likely to be obese, and you're able to go back 10 years in the medical records of a large outpatient clinic and compare those who took vitamin D supplementation with those who did not over 10 years in time, and you compare their BMI, their body mass index at the end of the time period. And you would calculate the results very much like you did in the prospective cohort study. It's just that you're actually collecting the data in a retrospective manner. Retrospective cohort studies are best when the data is collected via chart review, because asking people to look back over a long period of time and report on their exposures can be difficult, and sometimes they don't recall it correctly. The key points for cohort studies are as follows. Number one, there's no randomization in cohort studies. So when you study the association between an exposure and an outcome, you are unable to account for other potential confounders that might go along with that exposure. And this is really important. So for example, were the women in the hormone replacement study, the ones who are proactive enough to take hormone replacement after menopause, were they also just living healthier lifestyles in general? Were they getting medical care more regularly? Were they dieting and exercising and doing yoga and who knows what, right? And that's why they took hormone replacement therapy? We don't know. So you can imagine that you might have asked some of those questions, but you can't ask about everything. And the real kicker is that you can't ask about potential confounders that don't occur to you until later, or that aren't found to have an association until later. So for example, if we didn't know that statin use, cholesterol medication use, was associated with a lower risk of MI. And we followed all these women and didn't ask them about statin use. And then 10 years later, it was discovered that statin use was a major, sort of preventer of heart disease. Then that would be a problem because you hadn't asked about it in this cohort study, and you basically wouldn't be able to use the results. So cross-sectional studies are when you have an existing data set showing data at a single point in time, and you compare two groups, one that had the exposure and one that did not, on a certain outcome of interest. So for example, you want to see whether patients with public insurance are more likely to have schizophrenia than those with private insurance, and you have a snapshot of claims data from December 31st, 2013. And so you compare the group with public insurance against the group with private insurance, and you look at the number of people with schizophrenia in each group. Now, you can imagine that you might find, you probably would find that there's more people with schizophrenia proportionally in the group with public insurance. The problem with that is that it's actually a chicken and egg problem. The reason is that if you have a disabling mental illness, you actually qualify for public insurance, and this is the kind of study that wouldn't allow you to tell sort of which came first. You can also measure any variables that you think might be potential confounders. So here's your schizophrenia study. Just briefly, I'll say that the exposure goes on the left, the outcome goes on the top, and the positive of both should always be in the upper left corner. Again, you do an odds ratio by doing A times D over B times C. And so the odds of having schizophrenia, if you are on public insurance, is 2.7. So people on public insurance are 2.7 times more likely to have schizophrenia than people not on public insurance. This makes sense, of course, because actually the association is the other way around. People with schizophrenia are more likely to qualify for disability on the basis of their psychiatric issues, which means that they're more likely to have public insurance. The key points with cross-sectional studies are, most importantly, you only have data from a single point in time, so you literally do not know which came first, the chicken or the egg. You can only measure those confounders that occur to you ahead of time, like cohort studies. You're limited to people describing people captured by that data set. So people who aren't in that data set, you don't have any data on them and you can't say anything about them. You're also limited to the way that your exposures and outcomes were captured in that data set. For example, if it's a survey and they ask the question in a certain way that doesn't exactly get at what you want, like that's just too bad, you're stuck with it. So, for example, you could ascertain schizophrenia in lots of different ways. You could say to patients, has a doctor ever told you that you have schizophrenia? Or you could say to them, do you ever hear or see things that aren't there? And both ways will miss some patients. Some patients will not remember that they were told that they had it. Some patients will deny that they hear or see things that aren't there. So, it's an imperfect question. So when you have these cross-sectional data sets, you have to think about how was the question asked in the data set, and is it really what I want to know? This is just a brief note about confounding. We'll talk more about this later in the next lecture. But confounding is when there's a third variable that's related to the exposure and the outcome, but it's not on the causal pathway. So the example that I always think about is if you're looking at the exposure as a high-fat diet and the outcome is coronary artery disease or CAD, then high cholesterol is on the causal pathway. People who eat a high-fat diet are more likely to have high cholesterol, and people who have high cholesterol are more likely to have CAD. However, high-fat diet smoking and CAD, smoking is not on the causal pathway, but it may be possible that people who eat a high-fat diet are more likely to smoke, and that people who smoke might be more likely to develop CAD. And if that's true, and I suspect that it is, then your study might show a false association between high-fat diet and CAD that's really due to smoking. And so you'd have to collect data on smoking as well. There are definitely ways to adjust for confounding in the statistical analysis that we'll discuss later, but the really important point with all of these studies, and when you're designing a study or you're reading about a study that someone else designed, the really important point is that you cannot adjust for confounding if you didn't think about it ahead of time. If you don't collect the data on it and think about how you're going to deal with it, you cannot adjust for it if you don't have data on it. So experimental studies, there's two main types that we're going to talk about here, randomized controlled trials or RCTs and quasi-experimental trials. So when would you do an experimental study? When you know enough about the issue to feel that there's equipoise. We'll talk about that on Research Ethics Day, but that basically means that the scientific community truly does not know whether A causes B, but it seems likely enough to put a bunch of innocent people through an experiment to see if that's true. That's kind of like the cartoon version of what equipoise really is. There's a more elegant definition that I will tell you. when you have resources and funding to do it, because RCTs are extremely expensive to run. And when you need a definitive answer, these are considered the gold standard. So here's an RCT. You want to know whether taking simvastatin, which is a cholesterol-lowering drug, lowers the risk of myocardial infarction or MI. These are real studies that have been done, but again, I made up the numbers for the purposes of this class. So there are 10,000 subjects in your RCT. Each of them gets a pill they have to take every day. 5,000 get simvastatin, and the other 5 get placebo, which is a pill that has no actual medicine in it, or a sugar pill. There's 2 points here about RCTs. It's best to have the study be blinded, i.e. the subjects don't know whether they got the simvastatin or the other thing, but better still double-blinded, so the subjects don't know, and neither does the research team when they analyze the data. And you can understand now why there really needs to be equipoise. Like, you can't do this study today because we do know that simvastatin lowers risk of MI, and so it would not be ethical to take 5,000 people and say, we're giving you this placebo and not giving you simvastatin, even though we know that it would help you if you took it. And so you follow these subjects over time and keep track of who has an MI and who does not, and you generally use person time like you would in a cohort study. And so here's your results. It's really easy to compare the numbers in the two groups because they both have 5,000 in them, and you can already see that the MI rate is lower in the simvastatin group. But I calculated it 2 ways for you, by rate ratio and by odds ratio. Now, I didn't put the person time on this slide. You can do it that way too, and in lots of RCTs, they will show you the results by person time. If you're looking at the results by person time, you really can only do a rate ratio. If you have raw numbers like this, you can do it either way. As you see here, the rate ratio and the odds ratio approximate each other when your study is large and when the outcome is relatively rare, as it is in this case, but they are not exactly the same. The key points about an RCT is it all hinges on the randomization, how it was done, by what method. Was it a random number generator? Was it like everybody on Monday got assigned to this group and everybody on Tuesday got assigned to that group? You can think of lots of different ways to do it. Many of them inherently could have problems. You need to produce sufficiently similar groups via the randomization. And so typically in RCT articles, there's this thing called Table 1 that compares the intervention and control groups on a number of demographic factors. And really the point is that Table 1 should make it look like the two groups are exactly the same. In part because you want to know that the two groups are the same on the things that they measured, but if they're the same on all of those things, they're probably the same on unmeasured confounders as well. And this gets at the issue of what if 10 years from now some study is done that shows a really significant association between a confounder you didn't measure and your outcome. If you did an RCT, you're probably still okay, because if the randomization process was good, all confounders should be equally distributed, including unmeasured confounders. The other key thing about an RCT is how the endpoints, or the outcome of interest, were ascertained. You want to make sure that this method was correct and not likely to cause biased reporting. So when you think about the simvastatin and MI study, how was it ascertained whether someone had an MI or not? Did they ask the person? Did they go through their medical record? When they went in their medical record, how did they tell whether they had an MI? Did they use EKG data? Did they use lab data? Did they use data from cardiac catheterizations? You can tell there's lots of potentially different ways to do this, and there may not be 100% overlap between all the different ways. The other is that the people who are doing the ascertainment probably should not know which group got simvastatin. Because if you gave a bunch of cardiologists the results and said, figure out how many MIs there were in this simvastatin group versus this placebo group, and there was one that was kind of a gray area, like the EKG was kind of equivocal, you can imagine that people who had sort of a horse in this race might be more likely to call it one way or the other. So it's best if they're blinded. A quasi-experimental trial is very much like an RCT, except that the randomization process is different. Instead of randomizing individuals to blindly receive intervention or control, often groups are randomized to receive intervention or control, and they know. So it's useful in situations where it's not practical to contain the intervention to one person at a time. Recently, there was a smoking cessation program in public housing in Boston, and there was a lot of messaging in the elevators and bulletin boards and quick groups meeting in the downstairs, common room and whatever. And so what they did is they assigned an entire housing project to get the intervention and an entire one to get the control, because there's just too much potential contamination between an individual who's getting it and an individual who's not getting it but is reading all the bulletin boards in the elevators and stuff. So then when you're deciding who's going to be the control, is you have to pick another sort of entity, another housing project that's as similar as possible to the first one. I'm doing a project on women with intellectual disabilities who live in group homes. We actually randomized one whole state to get the intervention and another whole state to get the control, because there's so much overlap between social service workers that go in and out of group homes that we didn't think it made sense any other way. We could talk more about this in class, but basically it's a way to do an RCT without individual randomization. And the outcomes are measured and calculated in the same way as for RCTs. So the key issues when you're reading an article with a quasi-experimental trial is that the article has to explain the rationale for why it's not an RCT. And there must also be effort to the data collection process to limit bias, which is the idea that if people knew they were in the intervention group, which they usually do, they'd be more likely to report the outcome. And there should also be a table 1, again, showing how similar the intervention and control groups were, just like in an RCT. So this is just a summary slide. It's just to kind of help you study and to briefly review things. It has the type of study, whether it's observational or experimental, how the results are generally expressed, and key issues to consider. These aren't every key issue that you can possibly consider, but it's kind of like the important ones that you should think of immediately when you hear about these different kinds of studies. And we'll talk about this more in class, but this is just for you to have for your reference. And I'm happy to be contacted with any questions. Thank you.